Meiosis occurs to form gametes. Here the stage interphase occurs in which pairs of homologous uncondensed chromosomes in the diploid replicate. The pairs of duplicated chromosomes form sister chromatids. From here, this first stage of meiosis, meiosis 1, begins. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. In early prophase 1, each chromosome begins to condense and align precisely with its corresponding gene on the other homolog. Next, the formation of a zipper-like structure called the synaptonomal complex holds one homolog tightly to the other. During this association, called a synapsis, crossing over of paternal and maternal chromatid DNA happens. The location of where this happens is called the chiasmata. Additionally, in prophase 1, centrosomes begin to move and spindle microtubules begin to form. The kinetochores attach to the microtubules, and then the homologous pairs, which are also called sister chromatids, then move to the metaphase plate. The nuclear envelope also disappears. This stage takes the longest of all the stages. The second stage is metaphase 1, during which the tetrads homologs align on the metaphase plate, with one chromosome from each pair facing each pole. In this phase, both chromatids of a pair are attached to kinetochore microtubules on one end facing the pole, and centromeres on the other facing the other homolog. In this stage, Mendel's law of independent assortment also occurs due to each pair of alleles segregating independently of each other pair of alleles. The third stage is anaphase 1. In anaphase 1, there is a breakdown of proteins that are responsible for sister chromatid cohesion, and the homologs and centromeres separate. The homologs separate and move towards the pole they are attached to, guided by the spindle apparatus. The sister chromatid cohesion still exists at the centromere, however, and the chromatids move as a unit towards opposite poles. In this stage, we can see Mendel's law of segregation as the sister chromatids, which contain the alleles for the genes, have separated and are now going to be in different gametes. The fourth and fifth stage, telophase 1 and cytokinesis, occur at the same time. In telophase 1, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome consists of two sister chromatids. For some species at this stage, the chromosome condenses and the nuclear envelope forms. In this stage is also cytokinesis. Cytokinesis is the division of the cytoplasm and helps form two daughter cells. In animal cells, like this one, a cleavage furrow forms, indicating the two daughter cells separating and emerging. In a plant cell, a cell plate forms. In these stages, no replication occurs. This concludes meiosis 1. In between meiosis 1 and meiosis 2, there is no chromosome duplication. Now meiosis 2 begins. Meiosis 2 begins with prophase 2. Prophase 2 is similar to prophase 1, in which both daughter cells, the spindle apparatus, forms. Once again, microtubules start to attach to each chromosome. In late prophase 2, each chromosome is still composed of two chromatids connected at the centromere and move toward the metaphase 2 plate. Next is metaphase 2. In metaphase 2, the chromosomes are aligned on the metaphase plate. The kinetochores of sister chromatids are attached to microtubules extending from the poles. The sister chromatids are not identical to each other due to crossing over in meiosis 1. The third to last stage is anaphase 2, which is again similar to anaphase 1. A breakdown of proteins that allow for sister chromatid cohesion occurs, and the chromatids and centromeres separate. The sister chromatids move toward their respective side toward the opposite pole as an individual chromosome. The last two stages are telophase 2 and cytokinesis. These two stages occur simultaneously. The cleavage furrow and nuclei form, and the chromosomes begin to decondense. The meiotic division of one parent cell produces 4 genetically distinct haploid daughter cells, each with a haploid set of unduplicated chromosomes. These 4 cells either become 4 sperm cells or one egg.